Okay, this is a quick one about candidate genes. So we're in the section thinking about actually identifying loci, or if you watched the QTL analysis video about identifying actual genomic regions that contribute to these quantitative traits that we're talking about now. So candidate gene analysis has the goal of actually figuring out the identity of the QTL. So the QTL analysis told us near this marker locus, there is a gene that influences the phenotype that we're interested in. Candidate gene analysis is actually trying to figure out what gene it is that's near that marker locus and what does it do. So there are a couple of ways you can approach this sort of question. So you're looking for an association between a genotype and a gene product, either the structure of the gene product, maybe the function of the gene product, and the phenotype that that causes. So maybe you know something about a phenotype, and you know something about a gene or a product, a gene product, and so you're trying to find the area of the genome that codes for that protein product. Maybe you know a place in the genome and you're trying to figure out what that gene does. So we're going to look at a case where we have done a QTL analysis and we have identified a region of the genome where we have a strong association between that region, that marker locus, and a phenotype. And then we're going to, we also already know some of the genes in that region and what they do. Okay, so we're going to look at, okay, a slide is missing. It doesn't matter. We are going to look specifically at the case of Crohn's disease in humans and the gene NOD2, which encodes a protein that influences the immune system's response to bacteria in the gut. So this sounds like something that might have to do with Crohn's disease. We know that it's also in the region flagged by our QTL analysis. So we know about this gene. So we're going to do a test to ask the question, is this particular gene associated with Crohn's disease? And the test we're going to use for this is called the transmission disequilibrium test, which is pretty much what it sounds like. When this, we're going to say, let's see, is this trait more likely to be present in affected offspring than by chance? Let's think about the logic behind this. Parents, let's say, let's talk about carriers. So parents that are heterozygous, we know that they're going to pass this allele to 50% of their offspring on average, right? If they only have 2 offspring, you know, odds, coin flipping, right? But on average, among all heterozygous parents, 50% of the time, they're going to pass this mutated allele to their offspring, okay? The transmission disequilibrium test says, is the prevalence of the mutated allele 50% among affected offspring or greater than 50%? And if it's greater than 50%, that suggests that there is an association between receiving this allele from your parent and having the disease, okay? So if this allele was not involved in the disease, we would expect it to be randomly associated with whether you have the disease. So maybe you have 100 children and 50 of them have Crohn's disease, and of those 50, 25% have the mutated allele, right? That would just, that would obey our basic Mendelian inheritance laws of probability. We wouldn't see a statistical association between having the mutated allele and being an affected individual. Okay, so transmission disequilibrium test here is a little bit of a gel showing us the individual's alleles. So you can see that the wild type allele is just a bit shorter, longer, it's heavier, it doesn't go as far than the mutated allele. So we can see our parents 1 and 4 are heterozygous, 1 copy of wild type, 1 copy of mutated. and our affected offspring, both of them are homozygous for this affected allele. Okay, so this suggests that there's an association between getting the allele and having Crohn's disease, which suggests that there's an association between being someone who has Crohn's disease and the allele, right? That the allele might actually be causing or contributing to developing Crohn's disease. Okay, so we wouldn't just look at one family, we'd have to look at a whole bunch of parents and all of their offspring and ask, is there a greater than 50% association between getting this allele from a parent who is heterozygous for it and having Crohn's disease? Right, so the results of this study show that there's a greater than 50% chance of having the allele, the 69% chance. So among the affected offspring, we would expect, among the affected offspring who have heterozygous parents, we would expect 50% of them to have this allele if there was no association. We see that 69% of them have the allele, which tells us that there is an association between this allele and developing Crohn's disease. It's not 100%. This tells us that there, as we would expect in a complex disease, there's more than one locus that influences this disease. Okay, but this looks like one that might have an influence. Right? So that's candidate gene analysis. We have to know something about a particular gene or a particular gene product, and then do some investigation, again, relating genotype to phenotype.